Hello future doctors, welcome back to my channel Quick NEET Botany. In this video, we are going to continue the discussion of hidden lines of NCERT from the chapter Molecular Basis of Inheritance. There is a line in NCERT, in prokaryotes such as E. coli, though they do not have a defined nucleus, the DNA is not scattered throughout the cell. Yes, it is not scattered throughout the cell. DNA, being negatively charged, is held with some proteins that have the positive charges in a region called a nucleoid. Prokaryotic nucleus is called nucleoid as there is no boundary around it. The DNA in nucleoid is organized in large loops held by proteins. And one more question here. Theoretically, how many such beads or nucleosomes do you imagine are present in a mammalian cell? Here I am taking mammalian cell example, human cell I am taking as an example. In human cell, there are 6.6 into 10 power 9 base pairs. And you know, the number of nucleotides in DNA of nucleosome is 200 base pairs. So 6.6 into 10 power 9 base pairs by 200 base pairs is equal to 3 into 10 power 7 nucleosomes. And here, the packaging of chromatin at higher level, it requires additional set of proteins that are collectively referred to as non-histone chromosomal proteins. At higher level, these are required, but at all level, in eukaryotes, at all level, not only at higher level, at all levels, both the proteins, histones and non-histones are needed. That is one hidden line. Histones are needed initially and also at higher level along with that histones, non-histone chromosomal proteins are needed. So at all levels in eukaryotes for the packaging of DNA, both the proteins are needed. And in transformation experiments, in transforming principle, there is a line. Here, during the course of his experiments, a living organism had changed in physical form. What is the change of physical form here? R is converting into S. R, if you look at that R strain under the microscope, it has a rough texture or rough coat around it, that is rough in texture. But smooth, S strains are having smooth mucus coat. So there is a clear change in the physical form. R is transforming to S. So the physical form of the bacterium is changing from rough to smooth. And there is a direct line, but we may skip this line. Prior to the work of Avery, McLeod and McCarthy, the genetic material was thought to be a protein. Yes, before these three scientists' contribution, people they thought that the genetic material is a protein. And if you see, these three scientists, they concluded that DNA is the hereditary material. Of course, other biologists were not convinced initially. Later on, experiments proved that, like Hershey and Chase experiments, etc. But initially, they concluded that DNA is the hereditary material. Protein is not the hereditary material. And down there is a question. Can you think of any differences between DNAs and DNAs? Yes, we can say. DNA is a nucleic acid. DNAs, it is a protein which is acting as an enzyme. You know that this enzyme is cleaving the DNA. That is enzyme. It is not a nucleic acid. It is a protein. Next. Can you recall the two chemical differences between DNA and RNA? Yes, we can recall the differences. Chemically, DNA is having a different sugar, that is deoxyribose sugar. It has DNA with deoxyribose sugar C5H10 O4 sugar. It is having this sugar and it is having thymine. RNA is having, it is having C5H10O5, that is ribose sugar and uracil. These are the two major chemical differences between DNA and RNA. Here one more line we can see, that is, because of the rule of base pairing and complementarity, both the nucleic acids DNA and RNA have the ability to direct their duplications. But RNA formation from DNA, where it is replicating? Actually, because of complementarity, not only in DNA complementarity is there between the two strands. Why? Because adenine is having the capacity to pair with the thymine as well as guanine with cytosine. Adenine double bond, thymine, guanine triple bond, cytosine, you know that. In RNA also the same type of bonding is there. They have the tendency to bind with each other. Adenine with uracil, guanine with cytosine, of course, but it is not double stranded. It is only single stranded. So complementarity is not needed. It is not expressing that function actually. So being single stranded, it is not forming its own copy. It is formed from the DNA. And if you see in DNA, in Griffith's transforming principle experiments, heat which killed the bacteria at least did not destroy some of the properties of genetic material. Which property heat did not kill? That is complementarity. Even though the DNA molecule heated up to high temperature, both the strands can separate from each other. Of course, denaturation occurs. But again, if we provide appropriate conditions, when appropriate conditions are provided, then both the complementary strands, they come together. If they separated by heating, they come together. They have the tendency to rejoin, to rebind because of that complementarity. Next, RNA can directly code for protein synthesis. Yes, if you see central dogma, DNA is not directly coding for protein. It is first forming RNA, then it is forming proteins. So, is DNA directly forming the proteins? No. DNA is forming RNA. This process is called transcription. RNA is forming the proteins. That process is called translation. So, RNA easily expressing the proteins. It is coding for the proteins. And the total protein synthesizing machinery has evolved around RNA. DNA, however, it is dependent on RNA for protein synthesis. It is not directly synthesizing the proteins. Via RNA, it is synthesizing. So, DNA being more stable, it preferred storage of genetic information. But for the transmission of genetic information from DNA to protein, for the transmission of genetic information from where to where? Not from one organism to other organism, from DNA to protein, RNA is better. Next.